Welcome to the Deep Dive. Today, we're unpacking a really groundbreaking research paper that was published in partnership with the Hormel Institute at the University of Minnesota.
Yeah, it's an incredibly exciting piece of work.
It really is. So, our mission for this dive is to thoroughly understand this massive nationwide Italian consortium. It's known as Apollo 11. We're going to look at how they are using artificial intelligence, specifically something called multi-omics, to just completely revolutionize how we predict lung cancer treatment outcomes.
Right, and it's a phenomenal initiative. I mean, we are looking at a fundamental shift here. We're moving from guessing what might work to mathematically proving what will work based on, the most granular biological data imaginable.
Exactly. And to ground this, let's think about how we usually view medical diagnosis. There is this expectation of mechanical precision, right? You break your arm, the X-ray shows a jagged white line, and the doctor just points and says, there it is, broken.
Right. It's binary, clean.
Exactly. It's clean. But then you step into the world of advanced lung cancer, and suddenly that X-ray machine is pretty much useless. We are looking at a diagnostic landscape that is entirely murky.
It's incredibly complex.
Right. So if you put yourself in the patient's shoes for a second, imagine you or a loved one is diagnosed with advanced lung cancer. The doctor prescribes a cutting-edge immunotherapy. For some people, this drug causes a miraculous long-term remission. For others, it does absolutely nothing.
Nothing at all.
And the truly terrifying part is, half the time, we don't really know why.
We don't. And that uncertainty is the ultimate bottleneck in oncology right now. In clinical terms, we are battling two specific hurdles here. First, there's primary refractoriness.
Right.
That's when the drug simply never works from day one. The cancer completely ignores it. And then the second hurdle is secondary resistance, which honestly might be even more devastating.
Because it gives you hope first.
Exactly. That's when the treatment works beautifully for months or even years. The tumor shrank, and then suddenly the cancer adapts and the drug just stops working entirely.
Okay, let's unpack this. If we're going to understand how AI actually solves this, we need to understand what exactly we're treating here. The paper focuses heavily on two acronyms, ANSCLC and ASCLC.
Right. So those stand for advanced non-small cell lung cancer and advanced small cell lung cancer. And those cutting-edge treatments you mentioned, they're referred to as IO, or immuno-oncology.
Immuno-oncology, got it.
Yeah. And unlike traditional chemotherapy, which is, basically a poison designed to kill fast-growing cells indiscriminately.
Right, it kills the good and the bad.
Exactly. But IO therapies are different. They're designed to remove the invisibility cloak from the cancer cells, so your own immune system can actually recognize and attack them.
That sounds amazing in theory. But if they only work for about half of patients, how do oncologists right now, like today, decide who gets the IO therapy and who doesn't?
Well, currently, the standard biomarker, the biological red flag they look for, is a protein called PD-L1.
PD-L1?
Right, programmed death ligand 1. To find it, doctors perform an IHC, or immunohistochemistry.
Okay, wait, what does that actually look like in practice?
So they take a physical slice of the tumor from a biopsy, they drop specific chemical stains on it, and then they literally look under a microscope to see how much of this PD-L1 protein is present. The general idea is the more PD-L1 you have, the better the immunotherapy should work.
But relying solely on that one single protein is proving to be, well, wildly inadequate, right?
Completely inadequate.
Because it's like trying to predict the weather for an entire farming season by just looking at a thermometer outside your window on one Tuesday in April.
That is a perfect analogy.
Right. Like, sure, the temperature is a helpful beta point, but it completely ignores the humidity, the wind currents, the atmospheric pressure, the soil composition. One single data point just isn't enough information to predict a highly complex system.
Exactly. The tumor microenvironment is essentially a highly complex ecosystem. And this brings us to the massive paradigm shift that's driving Apollo 11. For decades, medicine has relied on what we call hypothesis-driven research.
Meaning you start with a guess.
Right. A scientist has an idea, a hypothesis like, maybe this one protein is the key. They design a clinical trial around that one idea, test it very slowly, and see what happens.
But cancer biology is just too complex for that now.
Way too complex. And it mutates too fast for us to guess one variable at a time. So we have to shift to data-driven research, which is powered by artificial intelligence and deep learning.
So to visualize the difference for you listening, think of hypothesis-driven research as fishing in the middle of the ocean with a single line. You drop your hook, you wait, and you just hope a fish happens to swim by and bite.
Yeah, lots of waiting.
Exactly. But data-driven AI research is like turning on a massive real-time sonar system. It scans the entire ocean simultaneously, showing you exactly where every single fish is hiding, how fast they're moving, and even what species they are.
That's it, exactly. AI doesn't need to guess. It can process massive quantities of complex, heterogeneous data simultaneously. It can find these hidden biological patterns across thousands of patients that the human eye could never, ever quantify.
Okay, so let's do a quick recap of the first 5 minutes of our deep dive here. The core problem is that we have a 50% treatment failure rate in advanced lung cancer because our old tool, looking at just one protein, isn't capturing the whole picture.
Right.
And the new solution is unleashing data-driven AI, which naturally makes you wonder, if you were building a supercomputer to predict your own health, what kind of data would you want it to look at? You'd want everything, right? You'd want the most diverse, comprehensive data set possible.
Absolutely.
But historically, getting that data has been nearly impossible.
And that failure is largely due to how our healthcare infrastructure is actually designed, specifically something called the hub and spoke model.
Okay, let's break down why that model is essentially starving medical research of good data.
Well, in a traditional healthcare system, smaller, local hospitals in rural areas or towns act as the spokes. They treat everyday patients. But if a case is highly complex or requires an experimental trial, that patient is transferred to a major academic research center.
Which is the hub.
Exactly.
Yeah.
But the problem is, the vast majority of patients are treated at those spoke hospitals. So all of their biological samples, their blood work, their day-to-day clinical data, it just sits in local filing cabinets or basic servers.
It's just wasted.
Completely wasted from a research perspective. And that excludes everyday patients from contributing to scientific breakthroughs.
And this means our current medical AI models are horribly biased. They're basically only learning from a very specific subset of wealthy or urban people who live near or can afford to travel to these massive academic hubs.
Right, it's a huge blind spot.
And this is where Apollo 11 completely flips the script. They intentionally built a distributed network across Italy that specifically includes rural clinics and island territories, like hospitals down in Sicily. They are deliberately gathering data from diverse demographics, varying socioeconomic backgrounds, and different levels of healthcare access.
They are effectively democratizing clinical data. And to execute this on a national scale, they utilize a highly secure web platform called RADCAP. This allows those local hospitals to capture and upload data seamlessly.
But the true administrative genius of Apollo 11, I think, is how they handle patient consent. They use something called one-shot consent.
Yes, which is so crucial.
It's such a simple, elegant fix. Normally, a patient fighting cancer is exhausted, and every single time their therapy changes, they're handed a mountain of redundant legal paperwork.
It's overwhelming for them.
Right. With one-shot consent, the patient signs a single, overarching document. It covers the collection of their data and biological samples for any innovative therapy registered in Italy since 2010.
It entirely removes the bureaucratic fatigue for the patient, and it clears the runway for the scientists.
Okay, here's where it gets really interesting. Once that consent is signed, Apollo 11 begins constructing what they call a multi-level biobank. And the sheer density of the data they collect is staggering.
We are talking about true multi-omics.
Let's do a deep dive into this. We need to walk through exactly what this AI is looking at, starting from the macroscopic level all the way down to the microscopic.
All right, let's start with clinical data. They extract electronic health records, or EHRs. But to prevent the AI from getting confused by different doctors typing different shorthand notes, everything is governed by a strict, continuously updated data dictionary.
So it translates all that messy free text into standardized clinical codes.
Exactly. So everyone is speaking the exact same digital language.
Makes sense.
Sure.
Then moving one layer deeper, we have radiomics. And we're not just talking about your standard chest x-rays here, right?
Oh no, we are talking about high-resolution 3D imaging, CT scans, MRIs, and FDG-PT scans, which actually show the metabolic activity of the tumors.
So wait, you can't just throw raw images at an AI, right? How does the AI know what part of the scan is lung tissue and what part is cancer?
That is where VOI, or volume of interest, comes in. You have to literally draw a boundary around the tumor. Apollo 11 uses a fully automated deep learning architecture called unmet to standardize these 3D boundaries. It mathematically segments the tumor from the healthy tissue.
Wow.
Yeah, and to ensure the machine isn't making mistakes, expert human radiologists semi-automatically trace a subset of the tumors to double-check the AI's math.
Okay, so alongside the 3D radiomics, they also collect pathology data. They take the physical glass slides holding the tumor biopsy and scan them into high-resolution digitalized whole slide images, or WSIs.
Right, so the AI can zoom in on the cellular structure itself.
Got it. Now we cross the threshold into the omics, the fundamental biological building blocks of the patient.
Yes. We started the foundation. Genomics. They perform WGS, whole genome sequencing to read the entire DNA blueprint, and WES, whole exome sequencing, which specifically focuses on just the regions of DNA that code for proteins. They also map SNPs.
Okay, let's clarify SNPs for a second. A single nucleotide polymorphism is essentially a single-letter typo in your DNA sequence. We all have them, and they are what make us genetically unique.
Right.
Finding those typos can tell the AI why you might process a drug differently than the patient in the exact next room.
Precisely. Next, they extract cfDNA, or cell-free DNA, from the blood plasma.
I find this part so fascinating because usually DNA is locked tightly inside the nucleus of a cell, right? So how is it just floating free in the blood?
Well, because tumors are structurally chaotic. As the cancer cells rapidly grow and die, they basically burst open and fragments of their mutated DNA spill directly into the bloodstream.
Oh, wow.
Yeah, it's incredibly valuable because it allows us to read the tumor's genetic fingerprints through a simple blood draw without having to repeatedly stab the patient's lung with a biopsy needle.
That's a huge relief for the patient. So we have the DNA blueprint. What's next?
Next is transcriptomics. If DNA is the blueprint, RNA is the messenger that carries the instructions to the builders. They use a technique called cRNA-seq, single-cell RNA sequencing.
And what does that tell the AI?
It tells the AI exactly which genes are currently awake and actively sending instructions in any given cell.
Okay, blueprint, messenger. Then what about the builders?
After that, we look at the actual builders, proteomics, which maps the amino acids and proteins, and then metabolomics and lipidomics, which measure the metabolites and fats that the cells are actually using for fuel.
So they're essentially auditing the entire cellular factory. They look at the blueprints, the work orders, the machinery, and the exhaust fumes.
That's exactly it. And they don't even stop at human cells. They're also sequencing the patient's microbiota from stool and saliva samples.
Because the bacteria living in your gut essentially talk to your immune system, right? Like we are finding out that your microbiome heavily influences whether an immunotherapy drug will be absorbed or totally ignored.
Exactly. The density of information they are collecting is just unparalleled.
Okay, so time for our second mini recap. If we take a breath and survey this massive ball pit of information we just built, Apollo 11 has fixed the hub and spoke model. They're gathering diverse data from all over Italy using one-shot consent. They have a biobank of standardized clinical records, 3D radiomic scans, pathology slides, and an exhaustive microscopic audit from your genetic typos all the way down to your gut bacteria.
It's a lot of data.
It is. And if you're listening to this and putting yourself in that patient's shoes, the immediate elephant in the room is privacy.
Oh, absolutely.
With all these millions of data points from your DNA down to your gut bacteria, how do we actually share this data without violating your privacy? Under European GDPR laws, medical data is fiercely protected. If you gather millions of deeply personal data points into one massive server, aren't you just creating the ultimate target for a cyber attack?
You absolutely would be if you centralized it, which is exactly why Apollo 11 uses a decentralized security architecture called federated learning.
Okay, here is the mental shortcut to remember how this works. The data stays home, the math does the traveling.
That sums it up perfectly. Instead of the spoke hospitals transmitting all their sensitive patient files to a vulnerable central supercomputer, the central supercomputer sends its untrained AI models out to the hospitals.
So the AI goes to the data.
Yes. The AI enters the local hospital secure firewall, studies the patient data locally, and learns the underlying biological patterns. Once it's done, only the mathematical weight updates, the learnings, are transmitted back to the hub. The actual identifiable patient data never, ever leaves the local hospital.
That brilliantly protects the data from hackers. But wait, what if an external researcher at a university in Tokyo or Boston wants to study the Apollo 11 data to test a new theory? They can't access the federated network, right?
No, they can't. And that is where generative AI comes to the rescue. Apollo 11 uses generative algorithms to create synthetic data.
Synthetic data, like fake people.
Essentially, yes, they generate digital ghost patients. These synthetic patients possess mathematically identical demographic and biological patterns to the real Italian cohorts, but they do not correspond to any real human being.
Oh, it's like training a facial recognition AI using incredibly realistic faces generated by a computer. They look human, they have human proportions, but those specific people don't actually exist.
Exactly. So external researchers can poke, prod, and analyze the data set with absolutely zero privacy risk to the actual cancer patients.
That's incredible. But solving privacy is really only half the battle. So what does this all mean for the doctors? We have the other half of the problem, which is trust. This is the classic black box problem.
Right, a major hurdle in medical AI.
Let's say this hyper-advanced AI looks at your federated data, compares it to thousands of synthetic ghosts, and spits out a prediction. Do not give this patient immunotherapy, it will fail. How on earth does a human oncologist look their patient in the eye and say, we are changing your treatment because the computer said so? They just can't trust a black box algorithm with a human life.
Nor should they. The clinical community rightfully demands transparency, and that is why Apollo 11 mandates the integration of XAI explainable AI. Specifically, they utilize visual interpretability tools called SHAP feature attribution plots.
SAP.
Yes, SHE stands for Shapely Additive Explanations.
Okay, in plain English, SHE plots basically force the AI to show its math homework.
Precisely. When the AI makes a prediction, the SHAP plot visualizes exactly how it arrived at that conclusion. It breaks the decision down into percentages. So the oncologist can look at the screen and see, the AI is predicting failure because it heavily weighted this specific lipidomic marker, combined with a 15% influence from this specific rough texture on the CT scan's volume of interest.
Wow, so it builds a bridge of trust. The doctor can tie the machine's logic back to established biological science.
Right.
But wait, I have to challenge something here about the data itself.
Yeah.
You mentioned CT scans and MRIs coming from all these different local hospitals. If a major hospital in Milan is using a state-of-the-art, brand-new MRI machine, and a rural clinic in Sicily is using an older scanner from 15 years ago, won't the images look completely different?
They will.
Won't the AI just accidentally learn to predict which hospital the patient is at rather than predicting the cancer?
That is a phenomenal observation, and it is one of the most notorious traps in data science. We call those batch effects.
Patch effects.
Yeah. If you don't correct for them, the AI will absolutely cheat and learn the technical noise instead of the biology. To neutralize this, Apollo 11 uses a mathematical technique called combat harmonization.
Combat, like combating the bias. How does that actually work?
Think of it like having 100 different photographers take pictures of a person's face. Some use vintage Polaroids, some use high-end digital cameras, some have really bad lighting. Combat is like a mathematical photo editor that neutralizes all the different camera effects. It adjusts the contrast and resolution so you're purely comparing the person's actual facial features.
That makes total sense.
And alongside Combat, they use complex regression models, algorithms like Lasso and MRMR. You don't need to know the complex math behind them, just know their job is to brutally filter out any data points that are influenced by technical biases, leaving only the pure biological reality of the tumor.
Okay, time for our third recap. Synthesizing this whole technology stack. We have federated learning, keeping the data safely at home, synthetic data, creating digital ghosts for global research, and explainable AI using SHAP plots to force the algorithms to prove their logic to skeptical human doctors.
Right.
While combat harmonization ensures the AI isn't being fooled by an old MRI machine. So if you're listening, ask yourself, would you ever trust an AI with your cancer treatment if it couldn't explain its reasoning to your doctor? Of course not. That transparency is what makes this entire project viable.
It's the only way forward.
And the wild part is, this isn't science fiction proposed for 2050. This is happening right now, in 2025.
It is entirely real. The governance structure is already fully operational. They have established a General Assembly and a steering committee to act as the gatekeepers for the network.
How do they decide who gets to use it?
Well, if a scientist wants to run a query on the Apollo 11 data, the steering committee grades the proposal on 4 strict criteria: relevance, urgency, originality, and feasibility.
And looking at the data cutoff from February 2025, the momentum is just staggering. They screened 52 clinical centers across Italy. 32 were rigorously selected to join the network. 20 have already received full ethical approval, and 7 are actively enrolling patients as we speak. There are currently 2,020 patients locked into the REDCap system.
It's incredible progress. And with that infrastructure in place, they have launched their primary scientific objective: The first major query Apollo 11 is trying to solve is building a multi-omic algorithm that specifically predicts IO efficacy immunotherapy success in patients with a NSCLC, advanced non-small cell lung cancer.
And to do this, they are zooming in on the immune system, doing deep peripheral immune profiling. They analyzed 264 fresh blood samples. What exactly is the AI hunting for in that blood?
It is hunting for PBMC's peripheral blood mononuclear cells.
Which are what exactly?
These are essentially the specialized frontline soldiers of your immune system. They are sorting through T cells, B cells, natural killer cells, and FACS-sorted neutrophils. But they don't just count them. They need to know what state those cells are in. Are they ready to fight, or are they completely exhausted?
How do they figure that out?
To figure that out, they scan for specific CD markers or clusters of differentiation.
Okay, think of CD markers like military uniform patches or name tags on the outside of the cell. They tell you the cell's rank and current orders.
Exactly. And the researchers look at a very specific roster of these patches. They're checking markers like CD11B, CD66B, HLA-DR, CD14, CD15, CD16, CD117, and CD71.
That's a very specific list.
It is. By reading these molecular name tags, the AI maps the exact battlefield composition of the patient's immune system at the precise moment they begin immunotherapy. And fascinatingly, the early data is already yielding strong results. It's confirming that a specific profile of robust T cells acts as a powerful, positive prognostic factor.
Meaning, if your blood draw shows the right combination of these T cell uniform patches, the AI can confidently predict that you are highly likely to go into remission.
Yes.
To actually generate these predictions, the researchers are deploying specific machine learning classifiers. They mentioned algorithms like random forest, multi-layer perceptron, and XGBoost.
Right. And to understand how these work without getting bogged down in computer science, let's look at random forest and XGBoost. Think of a single decision tree, like asking one doctor for a diagnosis. A random forest algorithm is like putting 10,000 doctors in a stadium, giving them all slightly different pieces of the patient's chart, and having them vote.
So it aggregates their decisions to eliminate individual bias.
Exactly. XGBoost, on the other hand, is an algorithm that learns sequentially. It builds a tree, sees where it made a mistake, and then builds the next tree specifically focused on correcting that exact mistake.
And they don't just blindly trust these algorithms, do they?
No, not at all. They train the AI on 80% of the patient profiles, and then they test it blindly on the remaining 20% to see if it actually works. They judge its success using strict statistical scorecards.
Like the AUC and C index.
Yes. The AUC simply measures how well the AI classifies success versus failure. And the C-index measures how accurately the AI predicts patient survival over time.
Okay, final recap of the real-world progress and early immune profiling results. They have over 2,000 patients. They're profiling millions of CD markers on T cells and using advanced algorithms to predict survival. Knowing that a simple blood draw looking at your T cells might soon tell an AI exactly which cancer drug will save your life, doesn't that make the future feel incredibly close?
It really does. It is incredibly rigorous work. If we step back and connect this to the broader landscape of medicine, Apollo 11 represents a monumental tipping point. We are finally moving away from hypothesis-driven guessing and toward true data-driven precision.
It's long overdue.
But there are, of course, major challenges ahead. They have to secure sustainable long-term funding, maintain a highly complex digital infrastructure, and continuously manage the inevitable heterogeneity of data flowing from dozens of different clinics. But the framework they have built, democratized data collection, federated privacy, and explainable AI, is laying the absolute foundation for a new era of oncology.
It's just mind-blowing. We are rapidly entering an era where your future medical prescription might be written by an AI. Wait, let me rephrase that. An AI that was trained on the synthetic ghosts of thousands of other patients. The exact cure for your illness might not just be invented in a sterile lab. It might be discovered hidden within the invisible shared data of a stranger sitting in a rural hospital miles away. It completely changes how we think about the shared human experience of illness and recovery. Keep diving deep, and we'll catch you on the next one.